Thank you so much. Thank you so much, Alejandro, for the nice introduction, and it's great to be here. I really am happy to tell you about our work tonight. It's great to be in South America for the first time, and I had no idea it was such a beautiful venue, and I'm looking forward to a week of great science. So my abstract said I was going to talk about a story about the, this is better. About the LARP protein, and I am, but I'm going to start with a story about ribosome collisions and how they activate signaling pathways, and then finish with a little story about LARP. What unifies, I hope, these two different sections is that concentration really matters. I'm going to tell you about functions of the ribosome sort of beyond protein synthesis, not in its role in translation, but a role in signaling pathways as well as a role in its own homeostasis. And in each of these cases, I think concentration really drives these decisions for the ribosome. So I'd like to start with this image of translation in the cell. This is a cryo-EM tomography image from Liam Holt's lab at NYU. This is a slice of a yeast cell. In gray are the organelles, in yellow are some particles that he used to look at the viscosity of the cell. And colored in cyan are the ribosomes. And in this image, you can actually quantify ribosomes at 23 micromolar concentration and 20% the volume of the cell. So you can imagine that if you have this image of the cell, you can imagine that the ribosome as a signaling molecule would make a lot of sense. The cell cares a lot about what ribosomes are doing. And moderating the amount of ribosomes in a cell is a central feature of what the cell needs to do, is to determine how many ribosomes to make. So with that as an overview, my lab and others for a long time have been interested in this basic process of translation, which is how ribosomes get loaded onto messenger RNAs, elongate through open reading frames, reach stop codons, and make proteins. Of course, ribosomes don't always walk through messages unimpeded. And in fact, we know that when ribosomes encounter problematic sequences, these could be encoded sequences or environmental damage, such as the effects of UV damage or ROS damage to mRNAs. We know the ribosomes stop because they can't get through the damaged mRNA, and that there are a number of quality control pathways in place to deal with these problematic mRNAs. You can imagine the ribosomes that encounter a problematic sequence can't complete the synthesis of the nascent peptide, and incomplete nascent peptides are toxic. And so we think a lot of the function of these mRNA-specific quality control pathways are meant to deal with incomplete nascent polypeptides, and there are E3 ligases that deal with it. You can also imagine that synthesizing over and over again a defective mRNA would be problematic, and so mRNA degradation tends to be coupled to nascent peptide degradation. And finally, ribosomes stuck on problematic sequences need to be rescued, and that's how we started studying these pathways. For a long time, the field wondered how it was that the cell was sensitive to a stalled ribosome, because you can imagine lots of ribosomes in the cell might be slow or stalled. And in a beautiful set of work from Hanny Zayer's lab at Washington University, he discovered that it's in fact ribosome collisions that are the trigger that says we really have a problem here. The ribosome is so slow that the lagging ribosomes collide. And in fact, that presents a new interface to the cell that is a binding site for a key E3 ligase that ubiquitinates small subunit proteins and leads to the recruitment of this collection of quality control factors. Along the way, as we and others were studying these pathways, in a set of ribosome profiling experiments that we were doing in yeast, we realized that MAP kinases were activated under certain cellular stresses, and we began to wonder about whether colliding ribosomes might also be involved in triggering the activation of these pathways, since it was long known that slow ribosomes could trigger the activation of what was known as a general stress response pathway, or the ribotoxic stress response. So the early work in this field was done by a lab in Oregon by Bruce Magan, and they really understood a lot about how slow ribosomes might trigger general stress response pathways when these pathways were overwhelmed with problematic species. And what we brought to this problem really was some biochemistry and a focus on ribosome collisions. So in work published a couple of years ago, that was done in my lab by postdoc Colin Wu, what Colin found was that ribosome collisions in the cell, when they overwhelm the mRNA quality control system, lead to the recruitment of at least two kinases, one GCN2, which stands for General Control Non-Derepressible. It was identified by Alan Hinnebusch in beautiful yeast genetics focused on starvation and the response of cells to amino acid starvation. GCN2 is a kinase that activates, and then I should say, then ZAC is the second kinase. It's a MAP3 kinase. We found that both of these kinases associate with colliding ribosomes, themselves become phosphorylated as kinases do, GCN2 leads to the phosphorylation, or its activation leads to the phosphorylation of EIF2 alpha, which is a key initiation factor, which, when phosphorylated, generally blocks more protein synthesis so that the cell has time to recover. It also activates a transcriptional program called the integrated stress response, again, to help the cell to recover. ZAC is a MAP3 kinase and has long been known to activate at least two different downstream MAP1 kinases, P38 and JNK, and P38 has long been associated primarily with cell cycle arrest, whereas JNK is associated with apoptosis. So in our initial studies, our focus was primarily on an apoptotic outcome and, it doesn't matter. In this Stoneley paper, Ann Willis's group showed that there was also this contribution of cell cycle arrest. And I should say that Simon Becker-Jensen's group has also contributed to our understanding of ZAC. I want to show you a little bit of data first to just give you a sense for how we initially sort of characterized these features of the pathway. What we did is we could expose cells to either intermediate doses of drug, shown here on the left, which was pioneered by Hanny Zayer's lab, or to doses of UV. Both of these treatments we know lead to the accumulation of ribosome collisions in cells. You can imagine in untreated cells where ribosomes are elongating, they're mostly monosomes. At high doses of drug, an elongation inhibitor, all the ribosomes get targeted and get stalled. But at intermediate doses of drugs, the idea is that some of the ribosomes bind the drug and others collide, and you can amplify collisions in those cells. UV damage is known to cause, in addition to DNA damage, mRNA damage, and we've shown that that leads directly to the accumulation of collisions in cells. So I'm just going to show you a little bit of data to convince that both of these treatments yield similar outcomes, and that's shown here. Shown here is a Western blot with untreated cells, anisomycin-treated cells, or UV-treated cells. What we're looking at first is the accumulation of ubiquitination on ribosomal protein S10, which is a marker for collisions. You can see in the presence of anisomycin or UV treatment, there's the accumulation of ubiquitinated ribosomal proteins, so collisions are happening. You can then look here at the downstream pathway from ZAC, because you can look at these untreated cells where ZAC runs as a single species, but in the presence of anisomycin or UV, there's a huge smear up where ZAC is heavily phosphorylated on inductions of collisions. So that's the autophosphorylation of ZAC. You can also look at the downstream pathways and see activation of P38 and JNK in both cases. You can nicely see with ZAC inhibitor that those reactions are blocked in both cases, but not with GCN2 inhibitor. Conversely, if we look at the output from the GCN2 pathway, you can see activation of phosphorylation of EIF2 alpha in the presence of either anisomycin or UV, and it's blocked by GCN2 inhibitor, but not by ZAC inhibitor. So this is the sort of evidence we use to convince ourselves that these pathways were taking place in these two different regimes. At the time, however, when we published this first paper, we wondered how it might be that the cell would integrate the various responses. This is primarily a survival response, as is cell cycle arrest, whereas apoptosis is forcing cells to die. And so we imagined that there might be some sort of collision meter where the cells measure how many collisions are taking place and integrates these pathways in order to make an appropriate decision. And that's what I'm going to tell you about today, is how we've pursued this question. We really were unable to get a molecular handle on it in our first publication. So what I'm going to tell you about is the work primarily of a postdoc in my lab, Niladri Sinha. We've had a wonderful collaboration with Alban Ordero, who's a mass spec person at MSKCC, and Sergi Rijot, our colleague downstairs, who's a MAP kinase signaling expert, Jeff Lee in my lab, and Connor McKenney in Sergi's lab. So first, I want to convince you that the cell, in fact, does integrate that sort of information in order to make an appropriate response and not always lead to cell death. What I'm showing you here is a titration of UV dose, and I'm looking at the proportion of cells that are either happily dividing in gray, in G2 arrest, or undergoing apoptosis. And you can see with increasing dose, well, in the absence of UV, cells are happily dividing. But as we increase the amount of UV dose, there's an increasing proportion of cells that die. So the cells clearly are integrating that information and making an appropriate decision. So what Niladri wanted to do was set up for a large-scale proteomic experiment where the kinases are what's being activated, so we need to understand the downstream signaling pathways. And so what he had to do was establish some parameters for this experiment. So what he did first is sort of think about the time course downstream. What we decided to use was keratinocytes, which are naturally responsive to UV. We wanted to set up a time course where we followed the sort of maximal activation of these pathways. What I'm showing you is here's an image of how we can quantify colliding disomes. This is a sucrose gradient resolving monosomes from disomes in the presence of RNase that digests either free monosomes or disomes, which are resistant to RNase treatment. And you can see this is the disome peak in untreated cells. This is after we treat with UV, and then 1 minute later, you can see a really very substantial increase in the number of disomes and trisomes. And we can follow this over time for 1 minute, 5 minutes, 15, 30, 1 hour, 2 hours, 3 hours. And so you can see that the peak signature of disomes accumulates in this early time part of the time course, and the collisions become resolved, presumably by quality control machinery, out at 3 hours. We then looked at the activation of the various pathways across this same time course, and you can see here the activation of ZAC from 1 minute through 20 hours. And you can see the activation happens really very rapidly, as does P38 and junk activation. So that's right here. So the ribotoxic stress response is quite an early response. For those of you who are worried about DNA damage and thinking I'm ignoring it, what I'm showing you here is that the DNA damage effectors come up much, much later than the ribotoxic stress response, suggesting it's a much later response to UV damage. So we decided based on these data to use these early time points and then to follow the proteomic outcome from the ribotoxic stress response as conferred by UV. So Naladri took these epithelial cells, cat cells, exposed them to UV irradiation, and waited over a time course of 30 minutes and took time points, did phospholipeptide enrichment, and looked at the total proteome. This is the principal components analysis to convince you that the time points cluster nicely, suggesting the data were robust. And then what do you get out of this sort of experiment? Well, you get a volcano plot. What can I say? So this is the volcano plot from the 1 minute time point. On the x-axis is increase in phosphopeptides relative to untreated sample. On the y-axis is significance. And you can see a lot of dots. If we look in those dots, we can begin to see things that make sense. ZAC comes up immediately. P38 downstream of ZAC comes up immediately, and GCN2 comes up immediately. So it certainly looks like the ribotoxic stress response. We can look at subsequent time points, 1 minute, 5 minutes, 15, and 30, and you can see a veritable explosion of data. We can quantify 50,000 peptides in there, phosphopeptides, and clearly cherry picking is not the solution to that problem. And so we had to come up with some ways to dissect these data and extract meaningful information. What we clearly would like to do is to define kinase-substrate relationships, look over time at who's primarily activated, what is how do you match up partners and enzymes and substrates. And so I'm going to tell you a little bit about how we've done this. We've used clustering analysis to dissect the temporal evolution of different phosphopeptide species. And what I'll start by telling you a little bit about is how we've used kinase inhibitors and knockouts in order to define the downstream pathways, and how we've used kinase signature assignments to better define these. So I'll start by saying what Naladri did next to sort of sort out the mess of those volcano plots is kinase inhibitors. So we used several different ZAC inhibitors, a GCN2 inhibitor, a P38 inhibitor, junk inhibitor, and actually an ATR inhibitor. He also constructed ZAC knockout cell lines and GCN2 knockout cell lines to compare to the inhibitor data, and he picked a single time point. And from that, he was able to show that, these dots that emerge to the left now are those that we lose in the presence of the inhibitor, suggesting those are the downstream substrates of ZAC or GCN2. You can pick up some global things just from looking at these data. You can see that the ZAC program is large. MAP kinases hit a lot of substrates, and they're explosive, and there's lots of substrates. By contrast, GCN2 hits far fewer targets. It's a much more limited kinase pathway. I won't tell you anything more about P38 and junk, but there's lots of interesting stuff downstream that we think has to do with ribosome rescue and dealing with the collisions. The main thing, but what I should say is that we're still left with a terrible problem, which is lots and lots of dots, and we need to figure out how to say something about where they derive from. And a great tool came along, which is Luke Cantley's lab has done a beautiful job of defining consensus motifs for mammalian kinases. What this lab has done is they've purified 400 mammalian kinases, and they set up glass arrays with peptide motifs all over them, added ATP, P32 ATP, and then analyzed the patterns on these glass slides for each of the mammalian kinases. And in doing so, they defined consensus motifs motifs for each kinase. What that allows them to do is to go into our volcano plots with thousands and thousands of phospholipeptides and to identify likely kinases upstream of those substrates and to greatly simplify the volcano plots. So if we start with the ZAC data, this is the ZAC knockout data and the ZAC inhibitor data. The first thing I'll tell you but not show you is that these data are really very strongly correlated, suggesting that the genetics and the chemical inhibition are comparable. And what the Cantley analysis told us is it greatly simplified these data, and it suggested that, in fact, downstream of ZAC, the main programs that are activated downstream of ZAC are a P38 or MAPCAP program, which MAPCAP is downstream of P38, and a JUNK program. And that's exactly what I told you in the beginning. We didn't learn very much new here, which is that ZAC is upstream of P38 and JUNK. So that was reassuring and suggested the methodology was working and was identifying appropriate pathways. The GCN2 data ended up being quite a bit more interesting, and I'll just start by saying that the phospholipeptides that are lost in the presence of GCN2 inhibitor include GCN2 itself, as well as several known downstream substrates, but it's clear that there's a very limited pathway downstream of GCN2. What was quite interesting, however, in the GCN2 data was what showed up on the right, which are things that increased in the presence of GCN2 inhibitor. So these are things that are normally negatively regulated by GCN2. And when we do the Cantley analysis on these data, what emerges is the following. There's a huge set of, or a nice collection, I should say, of species here that group together as downstream of mTOR. These are known mTOR substrates. And this suggested, therefore, that blocking GCN2, in addition to blocking the phosphorylation of this and its downstream role, involves that GCN2 normally plays a role in, at least with some distance, in regulating mTOR and in blocking mTOR. And this made a lot of sense because what phosphorylating eIF2 alpha usually does is it blocks ongoing initiation. What mTOR does is it's a very important regulator of initiation as well, but through a different route. And so what this suggests is that GCN2 both blocks initiation through its phosphorylation of eIF2 alpha, as well as by blocking activation of mTOR and thus preventing further loading. So by two different pathways, GCN2 is communicating that no longer should initiation take place. So in the presence of GCN2 inhibitor, you can imagine what's happening here is that there's hyperloading of ribosomes on these messages to increase collisions. And so you might imagine in this case that ZAC would be in fact hyperactivated, leading to activation of these pathways. And in fact, that's also what the Cantley analysis said, is that in fact these pathways downstream of ZAC are hyperactivated in the presence of GCN2 inhibitor, suggesting that one of the roles for GCN2 is to sort of slow down that ZAC activation pathway by blocking initiation. So I'm going to show you a little bit of data to convince you that that's true. We looked in wild-type and GCN2 knockout cells over time courses of UV irradiation. The first thing you can see in the GCN2 knockout cells is that there's a lot more of this ribosomal protein S10. That's consistent with the fact that TOR is well known to regulate ribosome synthesis for pro-growth. So that already made sense. You can see in the presence of UV that there are more collisions as seen through this ubiquitinated protein S10, this ribosomal protein that gets ubiquitinated. And in fact, you can see hyperactivation of JUNK here, which is this substrate downstream of ZAC, consistent with increased collisions and increased ZAC signaling. You can also see that over this time course, JUNK is kind of going up and then down. And at this point, we were already collaborating with our colleague Sergi downstairs, and what Sergi has is some beautiful tools to look at MAP kinase output. What he has is some optical reporters, which sit in the nucleus in untreated cells, and when they move out of the nucleus into the cytoplasm, they're an indication that JUNK has been turned on. These are kinase translocation reporters. So in untreated cells, you'll see that the reporter stays in the nucleus, but in the presence of either anisomycin or UV, the reporter moves out of the nucleus, and that allows him in real time to quantify JUNK activation live. So we use that experiment to characterize the response of our cells, our wild-type cells, to UV. So you can see first you get this nice peak of activation of JUNK downstream of ZAC and the decay of the signal. And you can nicely see in GCN2 knockout cells, you both get a hyperactivation of the amplitude of the response, as well as constitutive activation that doesn't die, consistent with what the proteomics told us, which is that there was hyperactivation of ZAC when you allow more and more collisions to occur. And the prediction of these data is that there'd be more cell death in the GCN2 knockout cells because they're not controlling junk activity. And in fact, when we look at cell death in response to UV, you can see in wild type versus GCN2 knockout cells a huge increase in the amount of cell death under this regime. And that increase, we think, is the result of increased collisions in the GCN2 knockout, and it's completely blocked by a ZAC inhibitor and mostly by a junk inhibitor, but untouched by a p38 inhibitor. So I hope these data have convinced you that we've learned a little bit about how GCN2 is talking to ZAC in order to regulate this initial response to UV and to collisions and to moderate its response. GCN2 is sort of keeping it under control. The next thing I want to tell you about is how we looked specifically at ZAC using the same phosphoproteomic data. And in this case, we looked at the time evolution of signal to learn something about ZAC itself and its activation. So ZAC is MAP3 kinase. This is its primary sequence with a kinase domain and its N-terminus, a leucine zipper, a SAM domain, and then an extended, relatively unstructured C-terminal tail that we and others have previously shown. It's this tail that's responsible for binding to the ribosome. So when you delete the tail, ZAC doesn't bind to the ribosome. So when we look at the phosphoproteomic data, specifically at ZAC, what you can see is there are as many as 36 sites that we can quantify on ZAC alone. And you can see this is the time evolution of their signal by the heat map. What we can do is cluster them according to their sort of time evolution. And you can see if we look at this cluster here, this is a cluster of phosphopeptides that's relatively flat over time. And you can see these sites are distributed along the length of ZAC. The second cluster, cluster 2, is quite an interesting cluster. It peaks around 15 minutes and comes down around 30, where we know p38 activation is taking place, and this one looked interesting. More importantly, we see that this cluster is kind of all together here right near the ribosome sensing domain, and so we wondered if phosphorylation of that domain might be critical to its interaction with the ribosome. Finally, cluster 3 is a small cluster. It's here, it rises and stays up, and it's a small cluster, and you can see they're sort of distributed all over. So Nalandri proposed with these data to make some cluster mutants. So we took these as a cluster and made serine to alanine mutations and asked whether loss of sites for phosphorylation impacted the activity of the ZAC protein. He generated knockout cells and then put these mutants back in and looked at activity. I'm just going to show you this one assay. It's this activation assay with the kinase translocation reporters. And what you can see is, again, in normal cells, wild-type ZAC, you get this nice activation downstream of UV. If we make a kinase dead mutant or an activation dead mutant, you can see there's absolutely no activation. So this reporter is reporting on ZAC activity. You can see when we make a cluster 1 or 3 mutation, we really have no impact on the output. But we make that cluster 2 mutation, as well as an all cluster mutation, what you see is that there's nice activation, it's sort of normal activation, but that the signal doesn't die. So this suggested to us that something about phosphorylation of cluster 2 had something to do with turning ZAC off downstream of the activation. At that point, we remembered that Simon Becker-Jensen, our colleague, had identified a potential phosphodegron motif in that cluster 2 area that could be a recognition site for the SCIF beta TRCP phosphodegron system. And in fact, there was a consensus there for it. And so Nalandri wondered what he could do to sort of further that idea. And so what he did was he did an IP with ZAC, an immunoprecipitation with ZAC in either uninduced or induced conditions to see if we could learn anything about these factors that are involved in this pathway. Those data are shown here. This is in the presence of anisomycin over untreated. And what you can see is that ZAC becomes more associated with beta TRCP, a known adapter for this system, downstream of collisions. So it's not initially associated with it, but downstream of collisions, it binds to this factor. You also notice on the left a lot of ribosomal proteins dropping off, consistent with the idea that ZAC might be on the ribosome, but might leave the ribosome downstream of these signaling events. So that led to the following model that ZAC might become activated on colliding ribosomes, leading to its dissociation, maybe as a function of that phosphorylation event. And that once off the ribosome, it might be degraded by this colon ligase system to diminish signal downstream of activation. So those were the two predictions, and I'll show you they both turned out to be true. This is a polysome profile looking at untreated cells, either wild-type ZAC, a kinase dead ZAC, or a cluster 2 mutant. What you can see is that ZAC, I haven't actually shown you yet, but that ZAC really does associate with elongating ribosomes. This is a Western blot of ZAC. It likes to hang out with ribosomes all the time. This is really associated. However, you can see that when you expose cells to UV stress, what happens is ZAC does begin to fall off ribosomes, possibly as a consequence of phosphorylation. The kinase dead mutant doesn't fall off at all, nor does the cluster 2 variant. So somehow phosphorylation of that cluster 2 is important for dissociating ZAC from the ribosome. This doesn't say it's degraded yet, though. This just says that the phosphorylation is responsible for dissociation from the ribosome downstream of collisions. Naladri went on to look at ZAC degradation downstream of activation of the pathway. He added anisomycin and simply looked at a Western blot over time, and what you can see is loss of ZAC. So in fact, ZAC is degraded. An activation loop dead mutant is not degraded. Cluster 1 and 3 mutants are degraded. That has no impact. But in fact, the cluster 2 mutant and the all cluster blocks degradation, consistent with the idea that that cluster 2 is in the area of the phosphodiegeron that's critical for recognition by that Cullen ligase system. And we have inhibitor data with MLN 194 to say that's the case. So the question we should all be asking now, though, is what is the value? Why would you want to degrade ZAC downstream of collision? What does that buy for the cell? How is that useful? And so we began to wonder if what that does is in certain cases, at sort of maybe intermediate doses of drug, the cell doesn't want to die, but it needs some time. It needs some time to regroup and to fix things. And that taking ZAC away prevents it being constitutively activated and activated and activated and leading to junk activation. And so we asked the question in the following way. We took cells and either pretreated them with just DMSO, or we added anisomycin and gave them a long time, a time period that would allow them to actually degrade ZAC. And then we asked after that period, are the cells less responsive to subsequent insults, and might that give them time to survive? So we did is after we took the cells and exposed them to these treatments, we then looked at the junk reporter downstream, and you can see with DMSO treatment, the null, what we've seen before, which is reasonable activation both by wild type and the cluster 2 variant, and slower, the cluster 2 variant's a little high. But what's striking is here, which is that the wild type cells, in fact, no longer respond to signal at all. Whereas the cluster 2 variant that wasn't degraded still is able to respond and still is constitutively activating this pathway. But the wild type cells, presumably, are somewhat protected during this period. They should be less apoptotic. And so when we look at the outcome of an apoptosis assay, a cell death assay, you can see, in fact, that that's the case, that wild-type cells are less apoptotic than cluster 2 mutant cells that are failing to sort of regulate and downregulate that ZAC signal. And the kinase dead is ignore that. So that's what we think the role of this degradation pathway is. So what I'm going to conclude this section by saying is I hope I've convinced you that we've come to understand something about how the cell is sort of monitoring collisions in order to activate different sort of outcomes. And I'm going to show it to you in the form of a model. Our basic idea is that there are 3 different regimes. In a first regime, when collision abundance is low and cells clearly are able to manage it and survive, we imagine that GCN2 nicely regulates and prevents further collisions from happening by blocking loading. There's very little ZAC activation, so ZAC doesn't get degraded. And in fact, junk activity never gets very high because there wasn't enough activation of ZAC to activate junk and lead to cell death. So that's the GCN2 regime and the survival regime where cells are activating survival pathways. We imagine there's an intermediate regime, a sort of tolerance regime, where ZAC does get activated and ZAC is degraded in some reasonable time period, that the initial insult wasn't big enough to cross some apoptotic threshold, and that this loss of ZAC gives the cell time to regroup and not over-constitutively activate and eventually lead to cell death. And so this is a protective regime as well, a tolerance regime. And finally, when the dose is super high of collisions, ZAC gets rapidly degraded, but it doesn't matter because it already passed this apoptotic threshold and the cells have no way to survive. They've already gone, the MAP kinase has exploded. And so we imagine that this is this third pathway. So there's the intermediate pathway where ZAC degradation matters, and this third one where apoptosis dominates. I had something to say here. Okay, so that's how I don't know what it was. So that's how we're thinking about that system. Oh, I know what it is. That ZAC, maybe in the end, ZAC is the collision meter. The ZAC's presence, its phosphorylation, its degradation, it's what's keeping track of how many collisions there are and what the output should be. So with that, I'll just transition and hopefully tell you a quick story. And this is a new story in the lab. It's brand new by a student, Jake Saba. And what this story is about how the ribosomes are sensed in cells by the LARP protein to regulate output or synthesis of ribosomes. And I'll just remind you about the abundance of ribosomes and how it's a major goal of the cell to keep track of how many ribosomes there are and how many it needs to make in order to differentiate and divide. Jake got into this project really with a general interest in, was interested in, let me start here. So a major way that cells regulate ribosome synthesis is through mRNAs, that all of which contain what's known as a top motif. A top motif starts with at the +1 with a cytidine residue and has 4 to 14 pyrimidines that follow it. All of the ribosomal proteins and many factors involved in translation have a top motif. These mRNAs are known to be coordinately regulated by a protein called LARP, which has at its N terminus a LA or a PAM2 domain, and at its C terminus a DM15 domain, which are responsible. The DM15 domain is known to bind to the cap of top motifs, and the LA domain is known to bind to poly-A tails. This protein is known to be regulated by the TOR pathway, so when cells want to grow, TOR turns on, and it certainly modifies the LARP protein, and that's thought to be critical for its activities. It's also known that ribosomes associated with these top mRNAs, sometimes it's 40S ribosomal subunits, sometimes it's the large subunit, but it's really not understood how the ribosome interacts with this complex and what the role of its binding might be. It's been argued that, in fact, LARP is repressive, but it's also been argued that it's not really repressive, it just allows a little bit of translation. So those are the questions we wanted to address. Jake began by a simple sequencing experiment where he fractionated cytoplasmic extracts over sucrose gradients to resolve polysomal RNAs, those associated with an ADS, a 40S ribosome, or free mRNA. So he simply sequenced across gradients each of these fractions to determine where all the messenger RNAs in the cell sediment, all at the same time. For example, a typical mRNA might look like this. This would be a well-translated mRNA that would predominantly sediment in the polysomal fractions with very little at the top of the gradient. So Jake did this initial experiment in 2 conditions: an untreated condition where cells are happily growing and in fact polysomes are relatively big relative to the monosome peak, or in the presence of taurin, which is an mTOR inhibitor that blocks polysomes and it shifts most of the ribosomes into an ADS peak. So it's grossly inhibitory. Then he looked at the distribution of all the mRNAs in the cell under these conditions. So if we first look at non-top mRNAs, what you can see is that this is thousands of lines here. This is where all the mRNAs distribute. They're largely translated. Most mRNAs are well translated. If you look at the top mRNAs, it's quite striking that tops, in fact, are biphasic. While there's a lot of top mRNA that's associated with elongating ribosomes, almost half of every top mRNA in this condition is wholly unloaded. It's sitting at the top of the gradient, and that suggests interesting regulation already. This was well known in the literature, that there's an on and off state of the top mRNAs specifically. When Jake treated cells with taurin, it's well known that taurin greatly regulates the top mRNAs, and you can see that here, that most of the translating top mRNAs are shifted into an untranslated fraction, whereas there's less of an effect on the top mRNAs. Well, what's really striking, if you look carefully at this, and was super exciting to us, is in the untreated cells, the top mRNAs really are all in this blue peak here, peak number 2, which is a 40S peak. And if you look in the taurin-treated samples, it's all in the ADS peak. So that taurin has sort of decreased overall translation, increased free ribosomal subunits, and now your tops are sitting in a completely different complex, in an ADS rather than a 40S complex. So it's quite striking. What Jake did is he confirmed this for a single top mRNA, and I'm just going to show you this because this is the assay I'm going to show you many more times. And you can see here, these are, again, untreated cells. This is now a single top mRNA in the blue peak, peak 2, and actin is way out here. And this is with Torin treatment. It moves from peak 2 to peak 3. So it's a very clear movement from peak 40S to peak 80S. So we were excited about this. It looked like an ADS complex. Is it a translating complex or not a translating complex? So we wanted to characterize it further. What Jake did is he began by spreading the gradient. So instead of focusing on this downstream stuff, he spread the top of the gradient over the whole 12 tubes. And those data are shown here. And now what you can see is in untreated cells, we now have multiple peaks for the top mRNAs. There's a little black peak here. That's a free mRNA peak. This is a 40S mRNA peak, and this is what we're calling a top ADS peak. And you can see, as we've seen before with Torin treatment, there's a great increase in that ADS peak in the presence of Torin. So this is now a spread gradient, 3 peaks. He first asked, is LARP an important part of this complex, as has been previously shown? He did that by using knockout cells for LARP1, and he re-examined those gradients. So this is what I'm showing you here. But now in the absence of LARP, you can see that you no longer form that complex, consistent with the idea that this top ADS complex has LARP. In the absence of LARP, you don't make the complex with top mRNA. The next thing, and this is actually the real slide, this is my favorite slide. As a ribosome person, it has salt titrations, it has everything. So Jake, what I've shown you so far is this top mRNA in an ADS peak in 100 millimolar salt. In the ribosome field, it's well known that real ribosomes that are really translating are resistant to salt treatment, and ADS ribosomes that are just kind of hanging out but not really translating are not. They're sensitive to salt treatment. So Jake increased the salt concentration and repeated this experiment, and you can see something pretty fun. You can see that in this higher concentration of salt, the ADS ribosomes are still there in the A260s. Most of the ribosomes in the cell are there. But that the top peak has made a really substantial shift to the left. It's moved. The top mRNA with its ribosomes have moved to the left. Consistent with the idea that maybe these aren't really translating ADS ribosomes, but salt-sensitive, not translating ribosomes. Jake went on to think about this in a couple different ways. He wondered what was in this top ADS complex, and the first thing he did on the right is he actually depleted small subunit proteins of the ribosome. And when you do that, when you knock down a small subunit protein, the cell ends up with more 60s than it does 40s, which you can see in blue here. And what you can see is that this top ADS complex with the top mRNA here can no longer form any complex with ribosomes. In the absence of 40s, it can't even make it can't make a 40S complex or this next complex. He knocked down a large subunit protein, and in fact, the top mRNA now moves to a peak where you have 40s plus the top mRNA, but not 80s, because you don't have the 60 to join. And then this is actually my favorite experiment. He knocked down an initiation factor, which is the last initiation factor involved in assembling a translating ribosome, but it needs an AUG. The 40S subunit is sitting in AUG, and the large subunit joins with this factor. And what he found is that the top ADS complex doesn't care at all about this factor that's responsible for only joining large subunits when there's an AUG. So this complex is not an initiation complex. There can't be an AUG there. It doesn't care. So that's what Jake got out of this sort of beautiful old school biochemistry. And at that moment, we connected with a sort of long-term collaborator's former postdoc, Jingdong, who actually had just solved the structure of the ribosome bound to a part of LARP. And that's what's shown here. What Jingdong at Fudan University had captured was a little bit of LARP found in the mRNA binding channel of the small subunit of the ribosome. He was able to capture 80 new amino acids that had never been seen before, and he was able to localize it to this region between these two better known domains. If I peel away some of the ribosome there, you can see it better. And if you were a ribosome person, you would recognize that this is hanging out in the decoding center of the ribosome and wrapping its way around the neck of the ribosome where the mRNA usually binds. It interacts with a couple ribosomal proteins, US3, US2, and ES30 in this area, but probably of more interest here is that if you look at other structures where there's an mRNA bound, you can see that the LARP protein superimposes on the mRNA binding site of the ribosome. So it doesn't just look like it's in the neck, it's right there. In addition, if you look at the structure of NSP1 protein, it's a SARS-CoV-2 protein that is known to block translation of the ribosome. It superimposes as well, as does SERPBP1, another known factor that interacts and blocks ribosomes in a storage complex. So these data, along with our biochemical data, unambiguously say that this is a repressive complex. There is no way that with LARP bound to these ribosomes, mRNA is bound, or you're going to initiate. This is a repressive complex. And so what it does is it allows us to put together this idea that there's these two domains, this domain that binds to the cap, this that binds to the poly A tail, and this little bit that binds to ribosomes. So Jake went on to confirm that the binding motifs that were found within this channel were critical. He did that by, again, using that LARP knockout strain, restoring either a wild-type LARP protein or a LARP variant that had mutations in that binding site. And Jake was able to show that in the absence of LARP protein, you don't make top ADS complex. When you restore wild-type LARP, you do make that complex. And when you use mutant LARP, you don't. So the binding motif specifies binding to the ribosome and formation of this top ADS repressive complex. So the final thing I'll tell you is we began to wonder if this binding of the LARP protein to the 40S subunit was going to be specific to the TOR pathway, where we had characterized it using taurin, and where most of the literature in the field suggests that the TOR pathway and its regulation of the LARP protein are really critical drivers of its activity. But we knew that there were lots of other things you could do to cells to increase free ribosomes, and that the cell wouldn't want to make ribosomes in any of those conditions. If ribosomes aren't translating, you don't want to make more ribosomes. And so this should be a broadly useful pathway. So Jake treated cells with a variety of agents that he knew would increase free ribosomal subunits in the cell. Sylvestrol is an inhibitor of initiation machinery. It looks just like taurin. Arsenite leads to stress granule formation, but is a totally different pathway. And puromycin releases free elongating ribosomes. All of these increase free ribosomes and free ribosomal subunits, and you wouldn't want to make more ribosomal proteins. However, what it was important to ask was, are these pathways also activating the TOR pathway, since people have always thought the TOR pathway is what's critical? What I'm showing you here are the key substrates of taurin, 4-EBP1. And you can see with taurin inhibition, there's really great downregulation of its phosphorylation. You can also see in this phosphotag gel that LARP is greatly dephosphorylated in the presence of taurin inhibitor. But with all of the other treatments that we did, they have no effect on the status of LARP phosphorylation or in the activation of the TOR pathway. And yet, when we look at the formation of this top mRNA complex, which I've shown you over and over, they all look identical. All of these treatments, independent of whether they've gone through a TOR pathway and the activation of that signaling, all of them lead to the formation of this repressive ADS complex, suggesting that in this case, we think that LARP is a much more general sensor of ribosomes and ribosome synthesis regulation. So that's where I'm going to close with this model, that we think that LARP is a general sensor of non-translating ribosomes. We've clarified a little bit about how this what was previously an ambiguous interaction of LARP protein with the ribosome, how it takes place, and it doesn't take place by the ribosome engaging with the mRNA at all, but rather with the ribosome engaging with this ribosome binding region on LARP to form these repressive complexes in a manner that responds to really all sorts of insults to the cell where you wouldn't want to keep making ribosomes. And so we think these are going to be a general principle. And in closing, I'll just come back to concentration matters. I think in both cases, this view of the cell as full of ribosomes, this is what we think about, but the cell is clearly full of ribosomes. And so the concentration matters. It matters for how many ribosomes load on a message and collide. It matters for how many ribosomes you want to make. And I think it helps highlight how we can think about different aspects of ribosome function cells beyond protein synthesis. And with that, I'm going to close and thank again Naladri as the postdoc who really led all of the effort on the signaling project in the beginning with the help of Jeff Lee, a student in the lab, and our wonderful collaborators Albin and Sergi and his student Connor. Jake led the whole project on LARP, and a great collaboration with Jingdong Cheng at Fudan and his students. And thank you all for listening.